making yourself fit.
All right. So what we're gonna end this unit, or this section of the unit, I should say, with is just talk about some more of the tools that we use in biology. So we talked about the microscopes, the compounding, dissecting microscope, which is what we generally use. But we need to know about some of the other tools. Not that we necessarily use many of these, but they're important to know a little bit about. So electron microscopes, they are similar in some ways to a light microscope, but also different in other ways. What do you think? What's the difference between an electron and a light microscope? Lauren?
Doesn't electrons send electrons through the thing that you're viewing, but a microscope or compound just sends light?
Yeah, you're correct in that light microscopes obviously use visible light to create the image. That's what the lenses, what's going on here? All right, yeah, so the electron doesn't use light, it uses a beam of electrons. We learned about electrons back in 7th grade, you know, very, very tiny particles with a negative charge. And because an electron is so small, it can create great resolution. Okay? Electrons were first developed in the electron microscopes were first developed in the 1930s. And they have some problems in terms of studying living cells because the objects that you're looking at in an electron microscope have to be placed in a vacuum. They often have to be coated in a reflective material, sometimes gold or some other metal. So you can't generally look at living cells in an electron microscope. So that's one of the problems. They can create great magnification. They can have great resolution as well because of very, very tiny particles. A small wavelength is useful in terms of resolution, much better than visible light. You know, the first electron microscopes were giant things. They look like this. They have a vacuum chamber. They have an electron source, and then they have a means of viewing those electrons as they pass through the image. Today they look more like a sort of computer with big tubes. This is a transmission electron microscope. And you know, I was looking earlier on eBay, you could buy electron microscopes. You could buy older ones, like $10,000.
Okay, yeah, sure.
So, you know, I mean, they're not millions of dollars. A brand new one might be, you know, $100,000. It depends on lots of different factors.
Can you buy one?
No. So the transmission electron microscope is similar to a compound light microscope in that the electrons are passing through the sample that you're viewing. Just like in a compound light microscope, light passes through the specimen. It still has to be sliced thinly. The other type of electron microscope is the scanning electron. And in the scanning electron, it works more like a dissecting microscope, where it sends a beam of electrons at the surface of your sample. They bounce off. Again, this is why it has to be coated in a reflective material. They bounce off, and then those electrons that bounce off are used to create a three-dimensional view. So this is a mitochondria of a cell. This is an image through a transmission electron microscope. Okay. This is an image of an ant through a scanning electron microscope, where it basically creates this three-dimensional view.
That's an ugly
Why does it kind of look like a person?
I don't know.
Like it has like a big nose or like that.
That's right. There's also a few other types of other microscopes. This is a scanning interferometric apertureless microscope, a SIAM microscope, which uses light, but with some special types of technology that give much better resolution than a typical light microscope. There's also something called a laser microscope, a confocal laser scanning microscope, which uses visible light but laser energy to create the image. And again, it can be used to view a specimen at different depths, and that image is created on a computer screen to give much more detail. Another common one is a phase contrast microscope. You read a little bit about that, but the phase contrast microscope works based on the principle that different parts of cells refract light differently. And those differences in refraction allow you to view different parts more clearly. So this is from the laser scanning microscope. These are images from a phase contrast microscope. So they give different sorts of views. There's also some other tools to talk about. When you read about the micromanipulator, that's a type of microdissection tool. These are very tiny tools that are used in conjunction usually with a microscope to manipulate very small things. And the different types of tools, there's a micromanipulator which can move things around. There's micro knives which can cut. There's micro pipettes which can suck things out. And this is often used in cloning and in vitro fertilization and things like that, where they can actually use these microdissection equipment to take a cell, puncture it, suck out the nucleus, and then inject it into a completely another cell. And that's how cloning works. They can use it to insert a sperm cell into an egg cell to fertilize that egg cell. So it's using these tiny little tools to manipulate cells. Centrifuge is, I thought I had one, Ms. Dowling has one. It's basically this tool that spins a sample around very, very quickly. What happens is different parts of the sample separate out according to their density. The most dense parts of the mixture end up on the bottom, and the least dense end up on the top. An ultra centrifuge is basically just a super high speed centrifuge that can separate even cell parts. So you end up with nuclei separate from mitochondria, separate from cytoplasm, etc. And you can look at those later. You can take them out and use them for various analysis. Tissue culture is just a way of growing a sample of cells of a certain tissue in the lab. Usually it's on a petri dish filled with a substance called agar, which keeps the cells alive. And you can basically grow, this is a micromanipulator. You could see what this scientist is looking through the microscope. The image on the screen shows what she can see. Here's a tiny little needle that she can use to puncture the cell here. This is a centrifuge. Put the samples here, go in test tubes here, and then it spins around very quickly. Okay. And that's what blood looks like after centrifuging, where you have plasma on top, red blood cells, and white blood cells and platelets in the middle. Yeah, it's just the color. And this is a tissue culture, where you're growing certain types of cells in the lab. And then the last thing is tools for separating molecules from each other. We're going to talk about some of these. Chromatography is a way of separating parts of a mixture according to the density and how they adhere to substance. And you do chromatography with paper. You soak a piece of special paper in a liquid that has various solutes in it. And as they rise up, like paper towel rises up as it works its way up the paper towel, the different solutes separate out. And they form a band. I'll show you a picture of that in a second. Electrophoresis works kind of in a similar way, but using electrical charges to separate molecules. Electrophoresis is used often in DNA or the amino acids from proteins. If if you've ever heard of like a DNA fingerprint, if you watch like CSI, they do those things, these DNA fingerprints, using electrophoresis. And then the last thing is spectrometry allows you to identify what's in an unknown sample. By basically burning it and looking at the types of light it produces. So this is chromatography, where you put a solution on the bottom of these strips of paper. It rises up, and as it does, the bands of pigments separate out from each other. We'll do some chromatography later on in the year. Here is chromatography of leaf extract from a plant. What happens is the various types of pigments separate. Different types of chlorophyll, keratin, and so forth separate according to how they adhere to the paper. This is electrophoresis equipment that separates molecules of DNA or amino acids, and it results in a banding pattern that you can analyze. And finally, this is a spectrometer, which you can put a sample of gas in there, and it'll tell you what percent is sulfur, what percent is oxygen, what percent is nitrogen. So cool.
So no homework for today?
No homework. We'll finish up with our last slide on Monday. And probably start a couple labs early next week. We have 2 labs to do.